बोल के।
हाँ जी ASP BMW है ना
हाँ
ये वाला
हाँ ओरल ट्रांसमिशन ठीक है।
हाँ जी
अगर कोई प्रॉब्लम होगी ना तो पहले ही बता देना, वो कभी कभी बड़ा लेट होता है।
नहीं ऐसा कुछ नहीं।
अच्छा कोई कह रहा था जब तक पता चला दे
वो थोड़ी दिक्कत, वो पैथोलॉजी वाले थोड़ा सिस्टम में घुस गए थे, अननेसेसरी उनको जरूरत ही नहीं थी, जो अपना लेक्चर देना है बस स्लाइड भी यहीं बनाने लग गए।
अच्छा
तो ये सेट फैसिलिटी ने जो अपना ये सिस्टम ये वो किया सारा, वो सारा वो हो गया था जिस वजह से हुआ।
अच्छा कैमरा है तो हम दिखते हैं स्टूडेंट्स,
आप जा रहे हैं और आपका ऑडियो वीडियो जा रहा होगा।
हाँ और वो साइड पे छोटी सी जो पिक्चर है जैसे बाकी याहू पे हो रहा है सेट का, क्लासेस भी तो हो रही हैं।
हाँ जी हाँ जी ठीक है।
माइक ऑन है।
हाँ जी बोलो।
कर दें स्टार्ट। हाँ AC तो है ना चल रहा। हाँ बस ठीक है। So good morning. We'll today talk of one important aspect of the training and knowledge of microbiology related to infectious diseases, and this is Antimicrobial Stewardship program. The objectives of today's talk is that at the end you should be able to state the problem of antimicrobial resistance, define Antimicrobial Stewardship program, it's also known as ASP in short, and that is the topic for today, enumerate the components and the steps of ASP, mention the role of ASP in containment of AMR, that is antimicrobial resistance. So as you are all aware now, at the end of your 1.5 years of microbiology and also knowledge of pharmacology and medicine, that antimicrobial resistance is a major problem these days, posing challenge in the treatment of infectious diseases, and it is emerging ever, and as more and more drugs are available, the resistance increases. So it is not only for India, but it's a global problem, and increased use of antimicrobial is one of the major issues. So it is both necessity as well as the disadvantage. So therefore, it being an international problem, at the United Nations, the global leaders committed to act on antimicrobial resistance in September 2016, and all member countries have made this a national priority, including India. So now in India also we have a antibiotic monitoring, resistance and control strategies. So what is the consequence? Why we are so worried about antimicrobial resistance? So besides, of course, mortality, morbidity increases, the duration of illness increases, there is high mortality, you have to treat with more expensive drugs, so the cost to the healthcare increases. So overall it increases the burden on the healthcare system because the patients are also admitted for longer duration. Then it negates all technological advances in the sense that lot of progress has been made in the treatment of cancers, in transplants. So while all these we are being successful in treating and increasing the life, it is finally the infection which becomes a major killer because it was multi-drug resistance. So the patients act as a reservoir of resistance organisms for the community also. So that's how it spills over to the community. It doesn't just remain in the hospitals. So why more antibiotics are used? Because healthcare associated infections are responsible for a large majority, bulk of the antibiotic use in the hospitals. Because in the hospitals it is, one is the patients are concentrated, they are getting treated, more antibiotics are being used. But also a lot of colonization takes place in hospitals because many patients are on devices. So the colonization also has to be treated with antibiotics. I'll show you in the next slide what is this colonization. Then we anyway have to use antibiotics because the immunocompromised host is increasing, more interventions are done, more indwelling devices are put. So you are likely to invade the sterile body sites in terms of these devices. You are treating an immunocompromised host. So these are all groups which are liable to infection with the opportunistic pathogens. Then lack of diagnostic facility in a large part of our country, because there only empirically everything has to be treated with antibiotics. So what is colonization? As I told you, as you can see in the picture, so in a hospital there would be some patients who are infected. You can see them as infected, you can identify them, but there is a much larger numbers which are colonized, means they are just the microorganisms stay on them. Mostly it is the devices because they form biofilms, so they stay on them and they serve as a source all the time, and many a time they would lead to culture positivity, and then you are not sure whether to treat or not to treat, and in the end you end up using antibiotics even in colonizations, which is actually not an infection. However, they serve as a source of infection. So this colonization rates are very high in tertiary care centers. These are all multi-drug resistance. They stay in the biofilms protected from antibiotics. So dilemma is to treat or not to treat, responsible for overuse of antibiotics, and this leads to a selective pressure. Now what is a selective pressure? This I'll just try to explain you with this diagram, that supposing there is one patient who is infected with a sensitive strain. Alright, this is sensitive to all available antibiotics, and you give such antibiotic, the patient gets cured of this particular bacteria, and what happens is some resistant mutants get selected. Well, that you know, that in a population there will always be a resistant mutant. So but they get selected out, others get killed, so it has no competition, it starts multiplying. And then another, so this set of organisms come which are sensitive to next generation of antibiotics. These you treat to kill these organisms, and another resistant mutant gets selected till such time. So this will be over the years, till such time now we have reached a stage where we have a situation where in a hospital our bacteria which is surviving in the environment is resistant to all available antibiotics, and this subsequently stays. It has an advantage. You are trying to kill it, you are not able to successfully eradicate infection. And this, so therefore this transmission, one of the most important reasons is through the hands of the healthcare workers, through the environment, through the equipment which is common use, and therefore we can control by taking care of this transmission from one person to the other person. Then the other thing why it disseminates so fast in bacteria, the resistance genes, is due to its rapid dissemination across the bacteria species, across genera, across organisms, that when there is a pool, as I showed you in the previous picture, that there is a pool of bacteria which has genes which are all sensitive and having resistant genes. When they get mixed in an environment, there is an exchange of DNA, and this exchange of DNA in case of gram negative is horizontally, so just across, horizontal transfer, very rapid spread, especially these ESBLs, carbophenamases, they transfer like this, so it's a very rapid dissemination. Parallelly also bacterial mutants are present in a population which are by virtue of the natural variations, and once you use antibiotics, these get selected out and then they try to survive in the atmosphere. So this is how the bacteria gets infection. This you have already read in your previous classes, but just to put it in the context of the present, antimicrobial resistance topic. So how do we save antimicrobials? No new antibiotic is going to come is seen across right now in the near future. So antibiotics, are a shared resource. This is the only drug where if you use in one patient, it has a effect and effectiveness in the others, as I told you, in terms of they getting selection of the resistant organisms. So this resistance management, stewardship, or taking care of the resistance is a behavioral issue, some biological, technical, economic, regulatory, educational. So it's a very complex aspect, and just treating a patient is only a part of it. So so we'll talk about that. Just giving you examples of some of the MDR pathogens. You must have read ESCAPE group of organisms, because these are the ones most commonly known phenotypes of resistance. MRSA, VRE, ESBL positive, carbapenemase producing Enterobacteriaceae, the MDR pen resistant Acinetobacter, Pseudomonas, colistin resistant gram negative bacteria. So these are some of the ESCAPE pathogens which are causing increased number of infection in the healthcare settings, with always a threat to spill over to community. So now we need to use the antibiotic judiciously. How do we do it? First is by having an antibiotic policy. Policy is more of a at the national level, management level, in the decision making. So India has a policy, each hospital should have a policy, and policy means the management will provide all commitment and support in the implementation of protocols which will take care of the antimicrobial resistance. So one limb of that policy is the antimicrobial stewardship, which is a day-to-day implementation to achieve, to be able to take care of the policy. Hospital infection control, as I told you, is another very important component, because as I told you, if the resistance, if the bacterial transmission is reduced, automatically antibiotic use will decrease. So now what is antimicrobial stewardship? So if you have to define, antimicrobial stewardship is optimizing clinical and healthcare outcomes, minimizing unwanted effects of antibiotics by reducing emergence of resistance mechanism, by reducing the selection of resistant pathogens, by reducing drug toxicity and adverse impact on patient safety. Because if they are to be treated with resistant due to resistant organism, expensive drugs, more hospital stay, it also has an adverse impact on the patient safety. So these are some of the goals, or you can say this is how you define antimicrobial stewardship. It's a complex program and you need a team for that. It has to be a microbiologist, pharmacist, infectious disease specialist, hospital epidemiologist or infection control practitioners, administrators, and computers play a large role in the data analysis and knowing your day-to-day for your day-to-day surveillance. These are some of the components of ASP. So let us just say, first of all, most important thing is data collection, target identification. So you have to know where are the problems in your hospital. So there you can focus your program. You can revise the formulary. So this is for those hospitals which have their in-house drugs, and so they will ensure what is available, what is not available, to prevent misuse. Then, and microbiology testing and reporting is very important because it also adds to your data. And also day-to-day modification of drugs. Education of healthcare workers, doctors, residents, very important. Restrictive policy is one limb where you can actually policy-wise you make a protocol that these such and such antibiotics will not be prescribed, maybe at the residents, only SR, or these at the faculty or HOD, like this. So that way you can restrict the use, or at least judiciously use antibiotics. Then ordering policy is also something similar to restrictive policy, formulary revision, so that you order those drugs which you have already discussed and considered as appropriate. Administration of drug in terms of dosages, mode of administration, the dosage, looking at the toxicity, PK/PD parameters. And one, they also say limit contact with pharmaceutical representatives because they tend to influence. So this was WHO in 2011 step-by-step approach to implement antimicrobial stewardship program. So how do you formulate for your own hospital practically? You develop the protocols and procedures for prospective audits and interventions. So here audit means you are actually doing surveys, you are looking at the data on the use of antibiotics. You do it in a prospective manner, day to day, and intervene as and when you think right things are not being done or judicious use is not taking place. Restricting the formulary pre-authorization. So for some drugs you can tell that okay, these only the head of the department or a senior faculty would just sign. You can make it easy by saying that okay, they can make a phone call and agree, but some kind of authorization is needed. Then one thing, because I have another talk to say, so I didn't spend much time. De-escalation is another aspect of stewardship, where you actually, once the patient comes who is very sick, you start with the high-end antibiotics. Then in 2 days you start reviewing all the data, all the laboratory reports, the culture reports, and then if you think this patient does not have infection with a very resistant organism, or based upon the sensitivity which should have come by then, you de-escalate, means you step down the antibiotics from the highest end to a lower level to which the organism is sensitive, and then also stop order. Sometimes you see the antibiotics which are written keep on being administered without anybody coming and bothering, so 14 days, 20 days. So stop orders should also be given at the right time. Dosage adjusting, parenteral to oral switch wherever needed. So these are the other aspects. So basically it means every day you must keep a watch on the antibiotic usage. That's why you have a big team, and pharmacists play a big role in this. Then cycling of antibiotics was suggested in some studies. It has really not been found very effective, but at least in some situations one can do that. Then of course education becomes very important, because here we are, so at your undergraduate level it's a kind of sensitization, but in postgraduates, amongst the other doctors also, residents, faculty, from time to time you have to go on educating on the problems of antimicrobial resistance and the requirement for antimicrobial stewardship program. But one thing I would say, that whatever is done, there should always be a feedback, because that only gives confidence to the practicing clinicians that okay, this kind of modality is actually helping the patients. So whatever your mortality, morbidity rates, length of hospital stay, antibiotic usage, cost reductions, these are some of the measurable data. So you generate this data, and after implementation you give feedback to the clinicians, to the management, so that you know in terms of some measurement that this is leading to some outputs which are significant. This is just to share with you how the Indian government, Ministry of Health and Family Welfare, is trying to cut down use of antibiotics. So on the antibiotics there is a red line, and never this should be purchased without the prescription, because you know antibiotics are available over the counter. So this was a program initiated by the ministry. So you see the logo, antibiotics used with care, and look for the red line. So as a responsible citizens, you should not buy, the chemist should not sell. So this is a responsibility of all the citizens. So to conclude, since we have no new antibiotics coming in the near future, we have to use whatever is available very judiciously, carefully, and we also understand that bacteria are becoming pan-resistant. So all available antibiotics, especially in the gram-negative bacilli. And with examples, you know, Klebsiella, Acinetobacter, Pseudomonas, Enterobacter, E. coli. So antimicrobial stewardship program should be in place for patient safety as well as to tackle the emergence of resistance. So, but we must remember, as I told you, no antibiotic policy will help in absence of a proper hospital infection control practices. So with this I conclude this lecture, and I think probably due to convenience this second one was clubbed together, but we can sort of link the two. So a proper infection control practices for stewardship program is mostly to do with the standard precautions in hand hygiene, in terms of the aseptic procedures. But one more aspect of hospital infection control is the biomedical waste management. So maybe because one thing was that this is a small lecture, it has been clubbed, so I have just tried to make a link of the two. We will now discuss biomedical waste management rules. I think by now, due to COVID, everybody knows this, so it makes it easier for me. But generally, to introduce the topic, biomedical waste management is within the legal framework. Constitution of India, Article 21, includes the fundamental right to clean environment with health and medical care for all citizens. And under this, biomedical waste management and handling rules were framed in 98. This is under the Environment Protection Act of 1986, and this came into effect from 20th July 1998. Subsequently, in 2016, it was revised, taking into consideration all the feedback and all the problems that were faced in the initial years of its implementation. So in 2016, it came under the Central Pollution Control Board, CPCB, which is Ministry of Environment, Forest and Climate Change. So it changed. So it is under this. Each state will have its own. In Delhi, it is Delhi. And this rule applies to all persons who generate, collect, receive, store, transport, treat, dispose or handle biomedical waste. So doctors also, hospitals, nurses, as well as the other end where the waste is treated. So all those people come under these rules, and it is their responsibility to follow these rules. The examples are hospitals, nursing homes, clinics, dispensaries, path labs, blood banks, health camps, forensic labs, research labs. So everywhere, you know, biomedical waste is generated. The key points of biomedical waste management is the segregation, which is the core. So it begins with segregation of the waste. And what we are worried about is segregate infectious from non-infectious waste. Then after you have segregated, collection and transportation between generation at the site to the treatment facility where they are going to disinfect this waste. And because all hospitals are not able to have their own waste treatment facilities, and also for standardization, common biomedical waste treatment facilities have been authorized. So this was this is now much more organized. The common biomedical waste treatment facilities, which collect waste from all these generating areas, take it to its treatment facility, and then treat the waste, which we'll see how they treat. And of course, the key point, most important, is all the staff, right from the generation to the treatment, should be trained in infection control practices. So this is the present segregation protocol. I think you are all aware because you see it in the hospital. Black is of course the general waste. This we will not talk, because what we are worried is not to waste our energy on every waste. We must focus on the infectious waste, because that is the purpose, to prevent the transmission of infection from within the hospital to the healthcare workers, besides of course waste minimization and appropriate disposal, also public sensitivity and misuse of some of the plasticware. So these are some of the reasons. So therefore let us focus on the infectious waste. The yellow bins, the yellow bags you must have seen, it takes care of human anatomical waste, some drugs, solid waste, expired or discarded medicine, soiled linen. The red bag is mostly for the plastic waste. So this is recyclable, that is why you separate it. Then there is a puncture proof, tamper proof container, which is for the wastes, sharps, so needles including metals. And then there is a, you can say, a blue or a cardboard box. It's a thick box where all glassware, broken, discarded, some medicine vials, ampoules, all these are put up. So again these are also recyclable, while this is non-recyclable. We'll see how they are all treated. So as I told you, you can put all these categories. So this is all available in the websites of the ministry. So once you put all this blood bag also goes there. Earlier this was not very clear, so all these go here. This is the soaked, blood soaked gauges, etc. So all this go into the yellow bag, and yellow bag goes for incineration. And what is important is this yellow bag is made of a plastic which is PVC free, so that when you are putting in incinerator at high temperature, the smoke which is generated or the fumes that are generated are not toxic in the environment. So these are the other drugs which we have labeled, just a pictorial, all laboratory waste like cultures, etc. Chemicals, liquid wastes. Then red category, which is the largest junk, the plastic waste. All are the PPE, the gloves now, then other devices, catheters, ampoules, these vacutainers, syringes. So all the plastic, which is a lot in the hospital, goes into the red bin. And this red bin can either be treated by autoclaving or microwaving. Hydroclaving is also a type of autoclaving. And then once that is autoclaved, it is subsequently shredded and can be recycled. The sharp container, as I showed you, the puncture proof, the tamper proof, can all is used for the, after cutting the needles, all the sharps, the needles and other metallic sharps can be put into that, and we should not use something like this which can be opened. Then the blue box that I showed you, all the ampoules and the injection, the broken glass, everything goes there. So to summarize, the color code yellow goes for incineration, the red bags for autoclave, microwave, subsequent shredding and recycling, the blue containers again shredding and recycling. Blue means the ones which has all the glass, broken glass, and the white container which is the puncture proof, again autoclaving, shredding and recycling. Then one aspect is the liquid waste. And so those needles can also be smelted and the metal be reused. One more thing is the liquid waste. The liquid waste has to be put down the drain in the running water, but each hospital or the facility should have a effluent treatment plant or sewage treatment plant within the facility, so that whatever leaves that hospital area and thrown outside into the community should be treated, and therefore nothing infectious goes out. Microbiology laboratory waste and blood bags are to be pre-treated on site. So others, as I told you, there is a common waste management facility, so you can send it to them. But the microbiology laboratory waste has to be treated on site, and all laboratory waste has to be treated at the level of the hospital. So each hospital should also have its, or the lab should have its own autoclave, or at AIIMS we have these microwaves where all the infectious in-house treatment takes place and subsequently is taken away by the common waste management facility. This you know, the general waste goes in black, but I think although that is not a hospital waste management topic, but you must also be aware that even now at homes solid waste management is being implemented, where you have to segregate blue and green. So if you not all, it is yet not become mandatory, but subsequently the recyclable will go into the blue and the other waste, kitchen etc., will go into the green. Right now black bags takes all the general waste. Only I would say one more thing, that in the context of COVID-19 there have been little modifications, because right now you all must know it, since we are in the middle of the pandemic, that with the quarantine facilities, isolation facility, COVID care centers, house isolation, home isolation, so there was a need to include some other areas outside a hospital, which was earlier not a part of the framework, which do not come under the biomedical waste handling rules. So there was a need, so that is why some revision was done, and mainly the other reason was the disposal of PPE, which was much more now. Quarantine and home care facilities, households were to be included, where generally the general waste, the black bags are there, versus biomedical waste where if the patient is under home isolation. Therefore sample collection units in the community are required, and that is done either by the common waste management facility or ULB, that is the urban local bodies. So they are authorized to carry, because you know the waste, the person who is taking these waste has to be trained and has to take care of the waste properly, proper disposal, it is his responsibility. Then in the hospitals they say much more stricter sewage treatment plant and effluent treatment plant monitoring should be done. Also initially all the food and all from the isolation facility used to go in the yellow bag, so which was then the numbers increased, incineration pressures increased, so there was a lot of load on incinerators. So therefore also need to minimize the yellow bag waste which has to be incinerated, because incinerators are expensive. So with all these there were some modifications. I will not go into the detail, but briefly in terms of the healthcare settings, what is important is you will do a double layered bag instead of single layer, which was being done all along. There are dedicated collection bins which are labeled as COVID-19. They are kept in the temporary storage rooms, and all the bags, bins, trolleys which you carry this waste are to be labeled as COVID-19 waste, and these should be properly recorded. There should be an audit. You have to ensure proper disinfection of trolleys and bags, the exteriors with the hypochlorite. Provide adequate PPE to the staff who is carrying this, from the hospital to the transport system to the treatment facility, and so all of them have to be educated and provided PPE, and then treat and dispose immediately on receiving. One should not store these bags for a long time. So Biomedical waste management is a legal responsibility. You must remember that we have to do it. There is no question of not being able to follow this or perform this duty. There is no reason to say I was very busy, so I left the needle in the treatment room, leave it to the sister. But the generator of the waste is responsible for the segregation. So please remember that. For you that is important because that is where you come into the picture. Rest is the administrative aspects. Only about 5 to 10% of the total waste is infectious. So focus on that 5 to 10% which is infectious. And so as a generator of the waste, you must make waste, proper segregation of waste a habit. Thank you. हो गया? पूरा हुआ? मतलब ट्रांसमिट हो गया?
मुझे
ये भी तो एक वो है, पता ही नहीं चलता कि वो हाँ कौन है. हो गया सारा ट्रांसलेट हो गया. कैसे पता चलता है?
ये सारे बच्चे
पता नहीं, बच्चे लोग आए भी, अब तो एग्जाम्स आ गए. अच्छा ये लोग जो प्रेजेंट है लॉगिन किए. अच्छा और ये का क्या मतलब?
ये तो वो आ रहा है
कुछ. अच्छा वॉइस का होगा. और वॉइस ठीक हो गई, ये भी कैसे सुनता है? वॉल्यूम ठीक किया, पता चल सकता है इसमें?
नहीं ठीक है.
ठीक है. अच्छा कोई नहीं, खत्म. आज तो सुबह की मीटिंग